This is my, is it recording? Yes, okay. So this is my bi-weekly for the week of April 29th, the week right before finals. So let's get to it. Okay, so in this bi-weekly, I'll mostly be talking about the Matlab script I made to go through the 10x genomics spreadsheet, pick out the heavy chain, the light chain, from the reads data for each of the clonotype, and just have them side by side for future assembly with the constant heavy and other necessary regions. So for ultimately homology modeling, I assume, in the obtainment of structures. So the script works fine. It samples most of the cases on the spreadsheet. It samples the standard. There's a heavy chain, and then there's a light chain of kappa lineage. That's a clonotype. It extracts both the heavy and light chain sequences for that. It also considers the case where there are two different light chains, and in that case, it picks the one with the most reads. But there's a few cases that I didn't include, particularly because there's so few of them. There's a case where there are two heavy chains and one light chain. I only, like, from skimming the entire spreadsheet, I only found like a few of those, so I don't know. But potentially, maybe I should include a criterion to select only chains that have above a certain number of reads. If they have below, I don't know, maybe like 1,000 reads, maybe they should not be considered. But either way, the script right now makes no such threshold. The only criteria it uses reads for is if there are two different light chains, it picks the ones with more reads. It doesn't like have a minimum number of reads that it goes by. So maybe that will be implemented in the future. The script it yields well over half of the sequences that are on the 10x genomics spreadsheet. And it's well over half because a lot of them on there don't even have a heavy chain. They're just a single chain, so they're basically useless. So yeah. So it works. It obtains the majority of the sequences. I don't know if that's too many or too little. That's an open question. I don't know. Whatever number you think is reasonable is what the script is going to be tailored to obtain. Okay, so the script only works for chimeras. The modification to make it work for the grafts is very easy, but I just don't want to do it because I don't really know where I can get the framework sequences for the grafts. I think it's not in the 10x genomics spreadsheet. I think it's somewhere else, but I don't exactly recall where. I know I get the CDRs from that spreadsheet. I don't know where I get the grafts, the frameworks from. And I realized on Tuesday that MIC is the culprit behind the long linker that supports the ligand in some of its docking. So MIC will need to be removed from all model structures. If that's what you want, yeah, because I believe it's also an area of low confidence as predicted by AlphaFold in it. Yeah, it most likely doesn't actually accurately represent what happens in docking. So yeah. Okay, so the Matlab script. It is here. This is the Matlab script that I wrote. I also have a slide for it here. Okay, so basically it opens the 10x genomics spreadsheet. I renamed it, and I didn't put all of the everything in the consensus annotations tab. I essentially only put the clonotype ID, the concatenated sequences, which can include the protein sequences for the frameworks and all of the CDRs. This is for the chimera case. The number of reads and the chain. That's essentially all I put. So it goes, it makes separate vectors of all of the different columns in the spreadsheet. MATLAB's not really friendly for importing it with this function read table. It imports it as a table, and indexing in tables is kind of difficult, at least for me, because I'm pretty bad at MATLAB too. So I made them into an array using the table to array function, and I converted them into a cell array so I can do like standard indexing with a for loop. So basically, the logic behind it is I went through all of the chains column, and I went through 3 different cases. So if there's first a heavy chain, a light chain, and then another heavy chain, in the overwhelming majority of cases on the spreadsheet, that corresponds to the heavy chain, and the first heavy chain and the light chain is a clonotype, and the third the third position is the beginning of a new clonotype. So I have in the first column I'll put the sequence corresponding to the heavy chain, and in a separate cell array, first row, I'll put the sequence corresponding to the light chain. This is one case. If there's a heavy chain, a lambda lineage light chain, and another heavy chain, again, these two will correspond to a clonotype, and then this is the beginning of another clonotype. So I just want to ensure that I'm covering each clonotype individually. So for the first row of a cell array of a cell array, we'll have the sequence of the heavy chain, and the second will have the sequence of the light chain. And then I considered another case in which there's first a heavy chain, kappa lineage light chain, another kappa lineage light chain, and then a heavy chain. These ultimately comprise the overwhelming majority of the spreadsheet. There were some cases where there were 2 heavy chains for a clonotype, but that's kind of difficult to encode. And also they were like a very, very small percentage of the whole spreadsheet where there are 2 heavy chains. There are also cases when there was only 1 chain, just 1 light chain, or just 1 heavy chain, and just each light chain was a differing clonotype, and that's useless. I mean, you need a heavy and a light chain. But yeah, this basically samples the overwhelming majority of the spreadsheet. So in the cases where there were 2 kappa lineage light chains, the most common occurrence, I made the sequences that went into the cell arrays be the ones that had more reads. So that's when I actually used the reads column. And at the end, I made 2 spreadsheets with the light chain and the heavy chain sequences side by side. And I have those spreadsheets. I'm going to pull them out right here. So these are all the clonotypes. This was essentially the spreadsheet that resulted from the code. So I have the clonotype ID, and then I have the heavy chain, and then the light chain. And the 0 just means this is clonotype 1, but 0 just means this is the position of the light chain, but the light chain is right here. So all the way going down. The gaps are where the criteria wasn't met, or there were just 1. For example, this whole swath of sequences, each of these clonotypes had only just 1 chain given for them, so I couldn't use them. As you can see, they're just listed once here. So yeah, it goes through the overwhelming majority of cases, getting the heavy chain and the light chain for each of the clonotypes. Oh, in this case, 1900 had 2 different light chains, and the one with more reads was picked. And I checked this. I checked this with the 10x genomics spreadsheet to actually verify that the proper chains were selected, and they were. And it goes all the way to the 9,000 something. Yeah. So it goes throughout the whole spreadsheet, and it gets the chains. So these chains will ultimately be concatenated with the constant heavy, the linker, and other chains to make the full length fab, which will then presumably go into homology modeling, or stability analysis, then homology modeling, and then yeah, the rest of the workflow. So yeah, this is the base for the entire sequence assembly work, I guess. So yeah, it's for the Chimera anyway. So yeah, it's here. Okay. Okay, so yeah, so questions going forward. Where in OneDrive can I get the sequences for the framework regions for the grafts? As mentioned previously, uh, the the CDRs, I know I can get them from the consensus annotation tab of the 10x genomics spreadsheet. Come on, load. Ah, there we go. Okay, so consensus annotations. So I know I can get the, yeah, so I can get the CDRs from here, from the graph CDR1, CDR2, etc. But I'm assuming I don't use these frameworks because only for the chimeras do I use the frameworks and the CDRs. So my question is, where do I get the frameworks for the grafts? That is my question. Um, so the script currently basically goes through the whole spreadsheet. If you want fewer clonotypes, do you want like a cutoff at a certain number of reads? Um, I don't know. I think I'd probably that would make it more manageable in terms of like the rest of the workflow. Because right now, I mean, some of the ones in the that the script selects, I mean, they're complete clonotypes, but they have like a very low number of reads, like in the hundreds, whereas others have reads in the tens of thousands. So I don't know. Maybe a threshold would be a good idea. I can't decide. I don't want to just make it arbitrary. Maybe there's something known that would make it like a better selection. And yeah, that kind of also goes over the third question. So how many sequences should we really want to work with? Initially, there's 4,672, and about 70 to 80% of those are sampled through the code that I made. Obviously, if we make a reads threshold, that will be reduced further. So I don't know. That's an open question. Okay, so yeah, so about Mick, um, I just want to ask this question again. We should remove it from all of the structures going forward, right? Because it's almost certainly an area of low confidence, as modeled by AlphaFold, and it also the docking in vivo won't be supported by Mick, so I'm assuming that we should just get rid of it, probably, hopefully. Okay, so what I'm doing now, studying for finals, but aside from that, when I have time, I'll go through and submit some sequences into AlphaFold as requested by Sam. Um, so the document he originally sent me and I thought that contained the necessary sequences, that was the document that on Tuesday was revealed to have the repeat sequences because there were two mix in there. And I can open it just for, yeah, this was the list of sequences, and each of these had two mix. So there's mic there, and then there's also mic there. And this whole thing is repeated there, basically. So that beginning part is repeated for each one, and there's two mix in every single one, which shouldn't be the case. So now, as you told me, I'm going to use, what is it? Oh, it's a different spreadsheet. This one. So this is the list of sequences, and there's only one mic. So I'm just going to use these. Learning glycan yeah, I should really get on that. And then figuring out the automation process. So the workflow is, I think, fully conceptualized. but it needs to be automated, and that might be a little challenging, particularly when we get to the design of ligand binding sites, the affinity maturation. But glycan-dock, automating it by itself doesn't seem that challenging because it's, from what it seems, it's not too much of a modification over standard protein ligand docking. And plus, like, the input is a protein ligand complex that you obtain. So it's not really, you don't need to automate like conformer generation, like params file generation, all of that. Like, you already have the complex to work with, so it's really just a matter of maybe making like an XML or something like that, like another docking script. The design of ligand binding sites protocol will be a little more challenging because I'll need a script that automatically selects the interface residues, and it also puts them into a res file. And because the res file is what tells the Rosetta design application, like, these are the residues you can mutate, and this is also how you can mutate them. You can mutate them with all 20 canonicals. You can mutate them with only one specific amino acid, whatever. I mean, the standard default is all 20 canonical amino acids. So that's probably going to be the most plausible one to use. And then I'll also need a script that conducts more than one round of design, because it's advisable to conduct more than one. If the models don't reach a certain energetic threshold, or I'll just conduct like 4 or 5 rounds of design and just call it good. I don't know. I don't know how many rounds of design are typically run. Usually not one, though. So if I do like 5 or 6 for all of them, hopefully that's good enough. And then I won't have to automate it. It'll just 5 or 6 rounds of design will be a given. I don't know. In order to decide on this, I'll have to look and see what others have done. Maybe I'll ask around on like ResearchGate or Rosetta Forum. But really, yeah, the difficult part will be the automation. Well, for me at least, because I don't have much programming background, even in Matlab. So yeah. Okay, so, and this is well, a very basic and crude flowchart, but it essentially covers my whole workflow. So initially obtaining the model GD2 from the 2D drawing, using OpenBabel, making it 3D, using BCL, generating 200 conformers, also generating params files so it can be used in Rosetta, obtaining homology models from AlphaFold. So I haven't really done this part. This is mostly Nathaniel. But no, I'm not going to include MIC in the homology models because it's problematic. I think ideally before the homology model step, stability calculations will also be conducted, and the structure will be optimized for stability either before or after AlphaFold. Then the structures will be relaxed into Rosetta force field. I've done that for the structures that I've docked already. Then protein ligand docking, standard protein ligand docking, fairly global docking, because I'm not going to be just in one coordinate. I'm going to sample like 3 or 4 coordinates on the CDR region, because I don't actually know where it docks. Best scoring, so the docking script currently selects the 10 best scoring complexes. I think I'm going to whittle it down to maybe like 3 or 2 or even 1. Glycan-dock will then be used on the best scoring complexes to refine the docked pose, to do some translations and rotations of the ligand in the docked complex to optimize its pose further in accordance with the energy functions that make it optimized for carbohydrates, because that's what glycan-dock is designed to do. Then the best glycan-dock poses are selected. Then based on those complexes, the residues that are, I don't know, like X angstroms away from the ligand will be selected for mutation for each structure, and then put into a res file. The res file will tell Rosetta Design what residues to mutate for affinity maturation and how to mutate them. I put X3 because, I don't know, for starters, I think I'll do 3 rounds of design. Maybe I'll do more. It seems a little complicated to, uh, I don't know, do a custom number of design iterations for each complex. Maybe some need more, maybe some need less. I'm definitely considering it. For now, I think I'll just do a standard 3 or 4 for each one. And then, obviously, I'm considering doing more. And then at the end, all of the sequences that meet a certain binding energy threshold will be extracted. And those will be the list of optimized sequences, really. So, yeah. And maybe after this step, they'll be verified for stability. I mean, they'll be verified for stability either before or after the AlphaFold step. And then at the end, I guess their stability will be verified again in case the affinity maturation mutations might have disrupted it. But it probably won't, because when I ran this affinity maturation protocol, it actually helped optimize the stability. It does that as well, so yeah. Okay, that was a long one. Sorry for the long, drawn out explanations, but the bulk of the matter here is the code and how it made this spreadsheet, including the heavy and the light chain sequences for almost all of the clonotypes in the 10x genomic spreadsheet. And then these will be concatenated with the necessary constant heavy, constant light, the rest of the fab sequence for the full length sequence. So yeah, that's the meat of the material. All right.